So the neuromuscular junction, or NMJ, is just another example of synaptic transmission. And that means that it builds on what we've learned about in our last unit. But it's the first specific synapse that we're talking about in our course. And so we'll need to describe it in just a little bit more detail. But the good news is that it follows the same patterns as we saw before in the previous unit. You have an action potential that leads to the release of a chemical messenger. The chemical messenger gives us a graded potential, and the graded potential gives us an action potential. The action potential then gives us contraction. So this is a zoomed in view of the neuromuscular junction with the alpha motor neuron at the top and the muscle fiber at the bottom. Again, we're only looking at one muscle fiber, but this would be happening for all the fibers within the motor unit. This whole process starts with the action potential firing in the motor neuron. That depolarizes the axon terminal, brings the voltage-gated calcium channels to threshold, and allows calcium to enter the axon terminal so that the vesicles can dock and the neurotransmitter can get released into the synapse. Now, since this is a specific type of motor neuron, the alpha motor neuron, it releases a specific type of neurotransmitter called acetylcholine. Acetylcholine can cross the synapse, the chemical messenger, and that means it will find receptors on the plasma membrane of the muscle fiber. The part of the skeletal muscle fiber that synapses with the neuron is referred to as the motor endplate. So we would say that the acetylcholine binds to acetylcholine receptors on the motor endplate. The acetylcholine receptor is a type of ligand-gated ion channel, and it opens when acetylcholine binds. This is a cation channel, which means that it's selective for positive charges. So when it's open, it allows both sodium and potassium to pass through it. But remember from talking about membrane potential that sodium's chemical driving force and its electrostatic driving force pull it into the cell. So even though the channel is selective for both ions, the major effect is actually that sodium enters the muscle fiber. This creates a graded potential. And if we were talking about a neuron here, we would describe this as an Epsp, because sodium entering the cell is depolarizing. But we don't use that term Epsp here for two reasons. The first is that there's only one channel, the acetylcholine receptor. So therefore, there's only one type of response that can be made here. In other words, we don't have the ability to produce any IPSPs, so it would be weird to use the term Epsp. But the other reason is that even though this is a graded potential, we always release enough acetylcholine to open enough acetylcholine receptors to give us enough graded potential to start the action potential. So in a healthy muscle, there are no subthreshold stimuli. These two things make this synapse a little bit different than the axodendritic synapse that we looked at in the neurophysiology unit. And that's why we can refer to this graded potential here at the motor endplate as endplate potential instead. But like before, the endplate potential diffuses away from its entry point into different regions of the sarcolemma, and that can initiate the action potential by bringing voltage-gated sodium channels to threshold. From that point on, the action potential inside of muscle is essentially the same as the action potential that's taking place inside of the axon that we talked about in our last unit. The only difference with the action potential is that this motor endplate is located in the center of the muscle fiber. So the action potential actually gets initiated in both directions and travels away from the motor endplate in both directions. Now, since there's no negative regulation or IPSP-like mechanism, the motor endplate can either be stimulated or not stimulated. In order to terminate the signal, we rely on the diffusion of acetylcholine out of the synapse or on an enzyme called acetylcholine esterase, which breaks down acetylcholine in the space, in the synaptic cleft.